Hi there, this is Mr. Rancor and
Mrs. Shelton.
And we are here to do activity 8 with you called Show Me the Jeans.
Ooh, I like Show Me the Money, but it's just the jeans.
Okay, all right, so I'm going to read the intro. We are on page 52 in the online textbook or page 41 in the paperback. Not that you really need to know that, but I just told you anyways. All right. So activity 8, Show Me the Jeans, reading. Here we go. Let me click out that X real quick just so we can keep the screen clean. Cool. All right. Since learning about Marfan syndrome, Joe has become curious about genes. He wonders exactly where they are and how they are transmitted from parents to their offspring.
Good question.
Even before he began his experiments on pea plants in the 1860s, Mendel knew that the genes had to be in the male and female sex cells. In other words, the sperm or the pollen for your plant and the egg. However, no one knew where in the cell the genes were located until 50 years later. An individual gene is too small to be seen with a light microscope. So how did the microscope enable scientists to figure out where in the cell the genes are located? Scientists studying dividing cells, like the one shown in the photo below, provided evidence to support Mendel's ideas. So we're talking about that one right there. So let's read the caption real quick next to that one. So it says the darkly, is that a word?
Mm-hmm.
The darkly stained chromosomes are lined up in the middles of the cells, getting ready to divide. Now we are on the next page, which I think was 53.
Correct.
Okay. So I'm on the guiding question. So the guiding question is the purpose of this activity. In other words, if you can answer this question by the end of the activity, then you've understood the lesson. So the guiding question is this. How do sex cells transmit genetic information for determining traits to their offspring? The first thing you're going to do is follow your teacher's instructions for how to use the stopping to think questions. You're going to go into the DNA Diner and you're going to access this document right here. It should look very familiar because we've done them before in several other activities. And you're going to answer using complete sentences, stopping to think questions 1, 2, 3, 4, and 5 after we've read it to you. Okay? Then be sure to think about how the diagrams relate to the text for each part of the reading. So that's your clue. You can't just read it and get all the information. You're going to have to read it, look at the diagrams, and see how the diagrams match up with what you're reading in the text in order to answer the question. So if you only do one, you ain't going to get the information. You got to do both. So this is what makes the lesson a little harder because now you're working with diagrams and text.
That means you're going to have to put on your thinking caps and think. All right, let's move on to the reading. Part A is all about chromosomes and cell division. And it says this: every organism must make new cells. Single-celled organisms such as bacteria, yeast, paramecia, and amoebas use cell division to reproduce asexually. In multicellular organisms, cell division, which is shown below, is necessary for the organism to grow to adulthood and to replace injured and worn-out cells. When you get a cut and you lose some blood, additional new blood cells and new skin cells are produced from the division of cells in your body. So if you take a look at this diagram, what you'll see is the parent cell. In the center of the parent cell is the nucleus. Okay, this, in other words, is like the main part of the cell, right? Okay, and then in this next part, you're going to see how the cell begins to divide, and then eventually those break off into two offspring cells. Okay, and then you'll see that those cells appear to get larger as they continue to grow. At this time, what we're going to ask you to do is to pause the video and complete stopping to think question number 1. Notice stopping to think question 1 asks you this: how is the function of cell division in single-celled organisms different from cell division in multicellular organisms? Okay, so if you listened carefully to what we just read about in part 1, at the beginning, it talks about single-celled organisms, and then about halfway through, it talks about multicellular organisms. And you'll see a diagram for the multicellular organisms that is shown below. Moving on now to the next part of the reading. You'll need to turn your page in your digital lab manual to page 54.
Okay. So, in the early 1900s, scientists studying cells in rapidly growing parts of plants made an interesting observation. They saw that just before cell division, the membrane around the nucleus was no longer visible, and the little dark structures, which they called chromosomes, appeared, as shown at the right. So this is what we're talking about. The chromosomes are right there. When the cells split apart, the chromosomes were divided evenly between the two new cells. When a cell is not dividing, the chromosomes are long, fine strands like very thin spaghetti, and they're packed into the nucleus of the cell. Before the cell divides, it makes copies of all its chromosomes. We've talked about that, so that should seem a little familiar about making copies. It makes copies of all its chromosomes so that its two offspring cells can each get a complete set. Again, that should sound like old information. You've already heard about that. Then the chromosomes become coiled, which makes them visible when under a microscope. Finally, the cell divides as shown below. And I'm going to scroll down so you can see that. Okay, so if you look right here, you'll see that here is the cell. The nucleus is intact, okay? In photo B here, or picture B here, the chromosomes become visible, and that's because they start to wind up, okay? And then C, the membrane around the nucleus is no longer visible. So you'll see here, you'll see the outline. Here it's starting to go lighter. And D, chromosomes line up in the middle of the cell. So this happens in every cell, every time a new cell is growing, okay? And then in E, chromosomes pull apart towards the opposite ends. So these separate into two single file lines. And in F, a new nuclei, nuclei is plural for nucleus, so two nucleus form in the offspring cells. And then finally, the offspring cells separate. Moving on to the next page, and we're going to page 55. So each cell in a human body contains 46 chromosomes. You need to memorize that number, 46 chromosomes, 4 and a 6. When a human cell divides, the resulting two cells each contain 46 chromosomes. We talked about that. You can't not have 46 cells or there's something wrong with your body, okay? We've talked about that, especially in my class. I know we have. How can 46 chromosomes become two sets of 46? It's not magic. Each crisscrossed chromosome is two identical copies that are attached to each other. As you can see below, each doubled chromosome then splits during division to become two identical but now separate chromosomes. And you can see that right there. So you'll see the single chromosome, it doubles and then splits into two separate chromosomes, making two copies. The next thing you're going to do is do stopping to think question 2. Remember, you're going back into this document right here. Okay, you're going to answer using complete sentences right here. And stopping to think question 2 says this: what would happen to the number of chromosomes in each cell if copies of them were not made before cell division? So think about that. You might need to go back into the text to look for clues and then answer it in a complete sentence right here. Go ahead and pause the video right now to answer stopping to think question 2. Pause.
All right, moving on to part B, chromosomes and sexual reproduction. The 46 chromosomes in a human cell can be sorted into 23 matching pairs, as shown below. Each chromosome looks identical to its partner, with one exception, pair number 23, which are also called the sex chromosomes. Okay? Female humans have 2 X chromosomes, while males have 1 X chromosome and 1 Y chromosome. Perhaps you think back to wellness. I think that you learned this in wellness, and you learned that the male, the sex cell, is what actually determines the sex of an offspring. Okay? So moving on to the next page.
Page 56. Got it.
Okay. Soon after the 23 pairs of chromosomes were observed, scientists noticed a pattern. When sex cells formed, the 23 pairs of chromosomes behaved just like the genes in Mendel's model. Sex cells, sperm and egg, remember sperm is the male sex cell, egg is the female sex cell, are formed by a special kind of cell division in which each cell receives copies of exactly half of the chromosomes. In humans, the body cells contain 46 chromosomes, but the egg and the sperm contain only 23 chromosomes. When a sperm cell fertilizes an egg cell to form the first cell of a new organism, the new cell has 46 chromosomes as illustrated below. Half come from the mother and half come from the father. So what you're seeing in this diagram here are sex cells and fertilization. So you start with a human body cell. A human body cell has 46 chromosomes, right? Now, when you're looking specifically at an egg and a sperm during fertilization, okay, we know the sperm fertilizes the egg. The egg has 23 chromosomes. The sperm also has 23 chromosomes. And when those combine to fertilize, to create the fertilized egg cell, the final cell ends up having 46 chromosomes. At this time, you are going to pause the video and answer stopping to think question number 3 on the analysis document that we've been sharing with you. The question asks this: why must the number of chromosomes in the sperm and egg be half the number of chromosomes in the other cells of an organism? If you rewind, I just told you the answer. All right, go ahead and pause the video and answer, and then tune back in for Mr. Rancourt.
And unpause. So now you just answered stop and think question 3, and now we are going on with the next section. Again, I am on page 56, towards the bottom of the page. When sex cells are produced, exactly one member of each chromosome pair moves into each offspring cell. Thus, when an egg cell and a sperm cell come together in fertilization, the new cell has 23 complete pairs of chromosomes, or 46 total chromosomes. It might help to think of the 23 pairs of chromosomes as 23 pairs of socks. Oh, I love this analogy. This is how I really got it. An egg cell has one of each kind of sock, and a sperm cell has the matching sock for each of the egg socks. Only when they come together, when the sperm fertilizes the egg, does the resulting cell contain 23 complete pairs of socks. One member of each pair has come from the female parent and the other from the male parent. The chromosomes of each pair carry genes for the same characteristics. See the diagram on the next page. But the two alleles of any one gene can be different, as you've learned before. 57, go. Here we go. Okay, so you see the chromosome pair here. You'll see an allele for a typical trait right there. Here's an allele for Marfan syndrome. And the pair of chromosomes shown is from a heterozygous individual with one typical allele and one allele for Marfan syndrome. All right, moving on to the next paragraph. So by comparing the microscope evidence with the work of Gregor Mendel, scientists realized that the chromosomes must carry genes. Once scientists understood the location of the genes and the way they were passed to offspring, they realized the importance of Mendel's work. Finally. So now what you're going to do, same routine, you're going to go back into this document, except now you're on stopping to think question 4. Here is that question, okay? Consider 2 children with the same 2 parents. Would you expect them to have the same sets of chromosomes? Explain why or why not. Or in other words, why are you and your siblings look different? Why do you look different? Well, you know. We want you to answer that right now. We ain't telling you that part.
All right. Go ahead and pause the video, answer that question, and then come right back. All right. Part C says this: so many genes. Human cells contain approximately 20,000 pairs of different genes. However, the human nucleus contains only 23 pairs of chromosomes. Each gene is a small portion of a chromosome. For example, the gene that causes Marfan syndrome is on chromosome number 15. Only by careful study can scientists determine which gene or group of genes is responsible for a specific trait, such as eye color or blood type in humans or seed color in pea plants. Furthermore, some traits are influenced by many genes. Environmental effects on the action of genes make things even more complicated. Some diseases, such as some kinds of cancer, are likely to result from the combined effects of several genes and environmental effects. The genes are part of a long molecule called DNA, which stands for deoxyribonucleic acid. Again, that's deoxyribonucleic acid. Each chromosome contains a long DNA molecule. Sometimes, before the cell divides, a mistake is made in the gene when the DNA is copied to make a new set of chromosomes. These mistakes are called mutations. If the mutation occurs during the formation of a sex cell, an offspring that results from the sex cell will be affected by the mutation. Genes give instructions to the body cells for making proteins. Even though some mutations don't make much difference, and some are even helpful, many mutations are harmful because they change the instructions in the genes. So at this point in time, we're going to ask you to go back to your analysis document. This is your final stopping to think question. And it asks this: how does a mutation change an organism? When do such mutations occur? Please notice there are 2 questions, which means you should have 2 sentences as a response, okay? 2 responses, 2 sentences to the 2 questions. All right? When you're finished with that, you're then going to move on to the analysis questions. There are a total of 3 analysis questions. However, you are only responsible for answering analysis questions 1 and 2. The following, let's see, what I'm trying to say is this. If we go here, you'll notice questions 1 and 2, you're going to answer here for analysis, and question number 3 is actually going to be on the scoring guide, so you need not answer it here. So again, you will know that this document is finished when you have answered stopping to think questions 1 through 5, as well as analysis questions 1 and 2. Make sure that you respond to each and every part of each and every question. Then go to your DNA Diner, find your scoring guide, and answer analysis question number 3 on your scoring guide. Mr. Rancourt, anything else?
Nope.
Signing off.
And we're pausing. And we're not.
Do not hit that trash can. I will